Well, I must say I'm excited about this next section, just as I have been for all of them. But genetics is very important to me. When I began in my studies of substance use disorders, especially alcoholism, if you had seen me in the hallway and said, you know, what do you think that there's a genetic influence here? Are you kidding me? I'd say this cultural, environmental condition, it couldn't possibly be genetically influenced. And indeed, it turns out that the proportion of the variants explained in severe repetitive alcohol problems is about 40%, and some would even say as high as 60%. So we are in a department that is very well represented regarding genetics work. And in this session, we're going to learn more about genetics of bipolar disorder from one of the first people to write seriously about genetics of that disorder, then substance use disorders in general, and then we're going to learn about autism, certainly a disorder that by its history you would figure must have a genetic component or would be very likely to have a genetic component. And there is indeed very interesting research going on regarding the potential for finding genes and finding genes related to that disorder. Our first speaker this afternoon is John Kelsoe, also somebody I've known and respected for many years. He's a professor in our department. He focuses on the genetics of psychiatric disorders, especially bipolar disorders. And I remember some of your earlier studies with the Amish, which I think were incredibly impressive. He uses a range of molecular genetic methods to identify specific genes, again, especially regarding a bipolar predisposition. John is also an important clinician who has helped out for years in our education program regarding residents, fellows, and medical students. And he is the director of the Bipolar Genome Study, which has 13 sites. His talk today is entitled Understanding Bipolar Disorder Through Genetics. So, John Kelsoe.
Thanks, Mark. I think this is really fun. I don't know about everybody else, but I'm really enjoying this thing, and I appreciate Mark and everybody else who put all the work into organizing it. I must have heard a dozen people say, wow, I didn't know he was doing that. So it's just really, really fun, and I won't go into my history with UCSD in great detail, except to say it's pretty much, my career's pretty much all been at UCSD, save 4 years at the NIH. I started here at 1982. I guess that's 33 years. So it's, I don't know, I'm proud to have been here for a significant portion of these 50 years. So, and most of that time's been spent studying bipolar disorder, and particularly when I was at NIH, the Huntington's disease locus was initially mapped by linkage, and it's suddenly just one of those aha moments of, wow, you could do this in psychiatry. And that sort of started my career, and it's been ever since. So just to say, to tell you, first of all, we have to be convinced that it is genetic, and twin studies have been a method time-honored to detect this, and I apologize, these are old data. There's some larger, bigger studies that they basically tell the same story. If one twin has bipolar disorder, and what portion of cases does the other twin have bipolar disorder, or a concordance rate, and you can see if you're an identical twin versus a fraternal twin, your rate goes up over fourfold. And in fact, with more recent, much larger data sets, the heritability of bipolar disorder is estimated at 80%. So this is really primarily a genetic disorder. Not entirely, but primarily. So here I'm going to, since I only had 15 slides, I mean, you just imagine the agony you put me through. So, but I thought I'd try and jam all of psychiatric genetics on one slide. That'd give background for everybody else. And so here we go. In the beginning, and I wrestled with whether I should put when darkness moved across the face of the deep. But in the beginning, in 1987, this is sort of the era of candidate genes. We had genes in mind that came about because usually they were targets of drugs. Serotonin, Prozac blocks the serotonin transporter, so therefore maybe there's something wrong with the serotonin transporter. Well, these are a whole lot of relatively modest to poorly powered studies, and I think it's kind of a broad consensus that not much really came out of that. Then we moved into the linkage era. Here the idea is that you look for, the important difference here is you're no longer looking at a single gene, you're looking at the entire genome with markers that will enable you to determine whether a certain portion of a chromosome is consistently being inherited along with the illness. And if so, you have linkage and indication that a gene for the disorder is somewhere nearby. Well, after 10 years of watching LOD scores after LOD scores, I basically came to the conclusion this was not really the right tool at the right time. It was a hammer when we needed a screwdriver in a way, in that the effect sizes for the genes that we thought we would find with linkage really just did not turn out to be there very much. Although, despite this, there were some big successes, most notably in schizophrenia with the discovery of the gene for neuregulin, which has led to ERB-B4 and a whole new area of biology-related illness, and dopamine amino acid oxidase activator, both of which, interestingly enough, were discovered by companies. So then we entered about 2005, the golden era of GWAS, because really we had been looking forward to this for a decade. A paper with Kathleen Marikangas, blanking, but had led us to think this, and it was just interesting as it developed. The resolution got higher and higher and higher from using 30,000 markers in the whole genome to now, not unfrequently, millions of markers. So this happened because of technology borrowed from the semiconductor industry and an ability to lay millions of little bitty pieces of DNA on a silicon chip. And each one of these little pieces of DNA, or oligonucleotides, is specific for a certain portion of the genome. If you have a stretch of DNA that's 20 bases long, that is a sufficient complexity to uniquely identify a spot in the genome. So in this way, DNA could be hybridized to these chips, and through a series of other manipulations, you would be able to measure millions of variations in the genome. Now, most of these were just an A turned to a C, maybe not even in a gene, and most of them were completely neutral and had no effect, but the hope was that they would be nearby a SNP, or single nucleotide polymorphism, that was causing the effect. So interesting experience in this. I've had the pleasure and honor to start and run one of these international consortia for the Psychiatric GWAS Consortium for Bipolar Disorder. And our latest paper is just coming out now. It's in press in Nature Genetics, where we've found 30 loci in a sample of what I think is about 18,000 cases and a comparable number of controls. So this kind of raises the initial question that we faced really 10 or 15 years ago, and that is the genome, are the hits half full or half empty? Is this really what we should expect? But as more and more it became apparent that the effect sizes of these individual variants were teeny weeny small, like 1.05, increasing your risk for illness by 5%. And that there were likely thousands of them involved, which was a classic polygenic model, as had been described by Sir Francis Galton and mathematicized by Ronald Fisher. Now, in comes genome sequencing. So here we can take it again one more step in resolution, where we go down from not being near the gene, but sequencing every single base every single of 3 billion base pairs in the entire genome. Or the sort of discount version of that, or exome sequencing, where you're sequencing all of the coding region in the genome, but not everything else. So this is very exciting. It's at the beginning right now, and it's clear that large samples are going to have to be accumulated, but it's very exciting. That's what I'm going to talk about now, as well as induced pluripotent stem cells, which is a revolutionary technology that has already completely changed biomedicine. So what I'm going to do is try and tell you about those things in my remaining 8 minutes. Okay. So just very quickly about this, the point I really wanted to make in this latest GWAS is that we found 30 genes, but the interesting thing is if you look at them, none of them would have been candidate genes. Not a single one of them would we have picked out and say this would likely be it. and the candidate genes are down in here. So that, I think, is very interesting. It illustrates the power of a genome-wide approach and sort of, in a way, an agnostic approach, and does what we really want in science. We found something completely different. So if you, I won't describe this in detail, but this is an analysis of the kind of categories of function and their relationship to each other that comes from that data I just showed you. And it's interesting, a lot of things you would expect from glutamate receptor related genes here and here, neural spine related functions. I mean, just about everything on here you can reasonably argue could be involved. Of course, that unfortunately is true with almost everything, if you have an imagination. So the one thing we've done in this regard is we conducted a whole genome sequencing study on our family sample, with the idea that the GWAS was really geared towards variants in the genome that are fairly common, and with the idea that each one contributes a small amount and you accumulate a whole bunch of them. At the other extreme is the idea that the variants are rare, but they have a larger sample size, and there's a gazillion of them in the genome to reach essentially a high level of heterogeneity in order to reach a population frequency. So it's in order to get at these lower frequencies alleles that we really required genome sequencing, which is what we did with Seth Ament and Jared Roach at the Institute for Systems Biology. And to make a long story short, we sequenced the entire genomes of 41 families, including 200 family members, and using some very cool statistical methods, mixed model methods, we were able to look at essentially association within families. And what comes out of this is really a replication of the GWAS results regarding calcium channels. From one of the earliest GWAS discoveries was this gene CACNA1C, and it's continued to turn up as the sample size get bigger, and in different studies, sometimes it's higher, sometimes it's lower, but it's always there. And when we looked at this family of channels, it was clear that this is the number of likely pathogenic variants in bipolars, and the blue lines number in controls, found in number of pedigrees. And similarly, over here we identified GABA receptors, GABA being an inhibitory neurotransmitter. The idea here really leads you to wonder, are these sort of promiscuous and allowing excessive neurotransmitter release and action potentials via calcium signaling? Or are GABA pathways perhaps defective and thereby disinhibiting the system? So, all right, that's where I'm going to stop on that one right there, but we'll kind of come back around to it. I'm going to switch tracks to the second topic, and that is lithium response, which leads to stem cells. So I've certainly been, after I began studying the genetics of bipolar disorder, because it at the time was regarded as the most heritable of illnesses. Now, I think that's really probably no longer the case, and those numbers have changed. But I also became interested in the idea that a subset of people with bipolar disorder, those who were lithium responsive, constituted a biologically distinct subset with a different mechanism of illness. So this is a study by Paul Groff in a Canadian sample where they took basically everybody who walked into the clinic and asked them their relatives with bipolar disorder, did they respond to lithium or not? And it was clear that relatives of lithium responders were more likely to also respond to lithium. Now, there's not a whole lot of data to support this, but we launched this gigantic study anyway. And so this was the Pharmacogenomics of Bipolar Disorder study, which was part of the Pharmacogenetics Research Network. And just to kind of go a little quickly, our goal was to get 600 subjects in what was a prospective trial, 11 clinical sites and 2 non-clinical sites for stem cells and genome-wide sequencing. You can see them up there. Basically, this is initially a relapse prevention design. We were to stabilize patients on lithium, watch them for a month off of all other meds, so they're on monotherapy, and then follow them for up to 2 years and look at relapse. Failures were to cross over to valproate, but I think a total of 6 patients chose that. So at this point, I'll skip a lot of the genetics because I want to talk about the stem cells. And this is work, we have a fantastic collaboration with Rusty Gage's group at the Salk. Those guys are just fantastic. Kristen Bernand, who's now at the Albert Einstein and Junyao, who went back to China, we chose a model that using stem cell technology, and again, let me back up. What this is enabled about 10 years ago, it was discovered by a Japanese group that 4 or 6 different transcription factors were turned on at really high levels in human embryonic stem cells. So they went about trying to use molecular biological methods to turn them off. And when they did that and tried it with enough different factors, they basically found, they basically found 4 factors which, when infected into a cell, will initiate reprogramming of that cell, which might be a fibroblast or a lymphocyte, and reprogram it to a stem cell. Once it becomes a stem cell, it is pluripotent and thereby can become any other kind of cell, in particular neurons. So this is the incredible excitement of this method, is that now we can have living neurons from patients with a disease, and the patient's still living too. That's the important part. So we did this for and did what Rusty called his favorite cell, this PROX1 positive dentate gyrus glutamatergic granule cell. It's an interneuron in the hippocampus in the dentate gyrus. And I think we all know why the hippocampus is interesting. We had small sample sizes necessary because of the really extreme laboriousness of this method, and this is what we found. Oh, sorry, these just, for years, all I had was numbers. I longed for colored pictures like the PET scan people had or other people doing my, so anyway, bottom line, these are just the cells progressing from untransformed into making their way to neural stem cells and neural progenitors. Then further along, they differentiate. So you have really differentiate into neurons where you see a more clear neuron morphology here, as well as expression of tubulin. So, okay, almost done. So here's the bottom line is this, that when we put an electrode in these neurons that came from bipolar patients, we found 6 out of 6 demonstrated a substantially increased rate of action potentials. So that's how the brain signals, is by firing action potentials, and that indicates a substantially greater signaling activity. So you can see this here, basically a whole bunch more spikes here than there are here, and this is quantified here with evoked potentials. Similarly for spontaneous potentials, which really showed an even greater difference here, and it's so visible you can just see it right here in the micrograph. And so if that wasn't enough, I have to back up to one say, you know, as someone who's treated many bipolar patients, it is tempting to say that the cells go fast just like the patients go fast. And who knows, there might actually be some kind of truth to that. But here's what was really the striking thing, and what validated this, I think, greatly, and that is that when we, of the 6 patients, 3 were lithium responders and 3 were non-responders. They were selected to be the end of the distribution. When we put lithium on their cells in vitro in the dish, what we found is a reduction in the firing rate back to normal, a rescue of hyperexcitability, but only in the patients who had had a clinical response to lithium. So these individuals were lithium responders, or a lithium responder. You can see the obvious decrease in rate that you can see right here quantified, but the non-responders, little change. So this was recapitulating the clinical drug response in a cellular model in vitro. And I don't know about you, but I think that's pretty cool. And so now this leads to all different possibilities to, if this is replicated and if this is real, the exciting thing is that this could potentially wind up in the clinic. Stem cells to fibroblasts to neurons is 6 months to a year if you're lucky, but it may be possible for us to identify genetic variants that are influencing that hyperexcitability, and someone could walk into the clinic, and as I like to say, I'm jealous of my colleagues in infectious disease. They can send a cup of spit down to the lab and get back a result that says ampicillin would be great, don't waste your time with gentamicin. I want that for our patients. This patient's going to do great on lithium, not very well on valproate. So this is sort of the dream that would come out of this. And again, probably by way of a DNA spit test, but maybe this stuff moves so fast. Who knows that there's not going to be a way developed to make stem cells in a week. Or something that would be in a way approximate for this measure. And lastly, So in genome-wide studies, we're finding things we never would have expected before. That lithium responsive illness is genetically distinct from bipolar disorder. We have a lot of other genetic data to support that as well. Drug response may be a powerful way to distinguish mechanism and to cleave the heterogeneity that exists in our various diseases. The stem cell neurons explain the hyperexcitable phenotype that was rescued by lithium in responders and not non-responders, and gee, I'd love to see this in the clinic. It's been a great time here, and this is just a really small portion of the wonderful people that I've had a chance to work with. Here on the left is the Pharmacogenomics of bipolar disorder. I've mentioned many of these. These are largely extramural collaborators, and some of these great folks here who are just instrumental colleagues, collaborators, friends, shoulders to cry on. And we appreciated them very much. Thank you very much.
I had a patient recently who she's 62 years old, never told us when she came down with what looked like a very anxious, depressive, bipolar attack at 62, that when she was 20, she had had a manic episode, was hospitalized, and responded quickly to lithium. She refused to go on lithium. I don't know what side effect specifically that she was objecting to, but she refused. She went through something like 8 months of pain, it almost broke up her family. Finally agreed to go on to lithium, and within a week she was significantly better. Within a month she was looking pretty much normal. So indeed, she was a responder, and the work you're doing is just magnificent. Second comment. What we've been hearing over these days is kind of like speed dating. That is, asking people to get to the exciting things that demonstrate something that they're most excited about now, and to try and do it in a way that people who don't do your work, that aren't in your lab, can really understand. And we've just had example after example after example of that, and yours certainly is among the wonderful examples.